Hello, my name is Michael Williams. I'm an assistant professor in the Department of Pediatrics and Human Development in the College of Human Medicine here at Michigan State University. I run a lab up here on the fourth floor of the Grand Rapids Research Center. My lab is focused on rare diseases, specifically those affecting the brain, in particular emphasis on neurodevelopmental disorders like autism spectrum disorder, epilepsy, intellectual disabilities. My interest in rare diseases really came from my passion for understanding how the brain works, and in particular, understanding how the brains of children work, and hopefully to better understand how brains develop and how brains develop differently in those with rare disorders, particularly certain genetic forms of epilepsy, autism, and intellectual disability. My real motivating factor here is to better understand how the brain normally functions and to help give families who are caregivers for those with intellectual disabilities or neurodevelopmental disorders a better understanding of the causes of these disorders, of what their medical future may look like, and of course, really our long-term goal is to ultimately inform therapeutic options. And we can't really do that until we understand how these gene differences relate to differences in the way the brain develops and functions. So my lab is really centrally focused on this essential question of how do rare variants inform how the brain develops. What rare disease families should know about my lab is that research has come a long way, but we still have a long ways to go. The tools that have emerged in just the past several years have dramatically accelerated how fast we can understand rare variants and previously understudied unknown genes impact the way cells like neurons in our brain work. We are leveraging these tools in my lab to come to a more detailed, more rapid understanding of how rare variants impact neurons, impact brains, and hopefully impact human life. In my lab, we run a basic neuroscience lab. And what that means is our real central goal is to understand how genetic variants in your families change the way cells function in my lab. And we can do this by investigating human cells in the lab or by investigating research subjects. My lab focuses on cells in a dish and on mice in a cage. But although mice aren't humans, we can put these same gene variants that we find in human populations in our model systems to better understand their impacts and their biology behind them. This is a really essential role because many of you have a long journey to understand what the genetic variants you carry are. Our goal is to help now that next step. What does that gene variant do? How does your specific variants found in your family or in your medical situation change the way that gene normally functions so that you can have some context for understanding these variants and so that we as a field can provide a better understanding and better therapeutics. If you're a student or a potential trainee who is interested in rare diseases, the first thing I would say to you is go for it. This is a really exciting field. It's a very exciting time to be in this field. It has become so much more feasible over my early career to sequence people, right? To understand what their genome is, what their transcriptome is, to understand what genetic variants they have and what transcripts they produce. And on the flip side, in the lab, our tools to manipulate the genetics have become much better, right? We are now in this era of CRISPR and other gene editing technologies. And it is now becoming routine to employ these tools in the lab. We can, as a matter of course, design tools to investigate just about any gene. and to recapitulate specific mutations found in the human population in our research subjects. So as long as there is sufficient interest on the trainee side, we can always try to bring new variants into the lab to understand them. And so these can make excellent rotation projects, summer projects, or graduate or postdoctoral or assistant professor careers. There is so much that we don't know about what these genes' basic function is, that we really need to address this gap. Now that we can rapidly identify variants, my feeling is we need to move more rapidly towards phenotyping them, describing what their consequences are, understanding their biology. This will also not only help us understand the essential biology of things like brain development, which is already pretty complicated, but also understand how rare variants and rare diseases of these genes and of these systems impact the way the brain functions. I think if we want to be able to understand a rare syndromic form of autism or a rare gene-driven form of epilepsy, we really want to be able to not just tell that patient what mutation they have in what particular gene, but follow that up with, here's what we have found out about this gene, here's how we know it functions, and here's what you might expect in your medical future, so that ultimately we can provide this next answer, which is, here are the specific therapies that we think will work best for you and your family. This is a field that needs you, and I encourage you to come and join us here in my lab or in any of these other labs here at MSU or our other research partners. So thank you. Thank you for your interest, and good luck.